Hi guys, welcome back to my channel. This is the part 2 video of the chapter 1, which is nutrition. So let us begin our video by talking about C.B. Van Niel. C.B. Van Niel, first he worked on the purple sulfur bacteria and found light plays specific role in photosynthesis. Here the purple sulfur bacteria, which is a type of bacteria which shows the autotrophic nutrition, mainly it shows the chemosynthetic type of nutrition, and that bacteria takes the H2S gas and it releases the sulfur from H2S gas, and that is a type of anoxygenic photosynthesis. It is called as anoxygenic photosynthesis because here the sulfur is released and not the oxygen. If oxygen is getting released during the photosynthesis, then it is called as oxygenic photosynthesis. Because here no oxygen is released, it is a type of anoxygenic photosynthesis. And H2S gas, which is the hydrogen sulfide gas. So, but again later the C.B. Van Niel, he worked on plants and he found that in plants the oxygen gas is released when H2O, that is the water, is used. That is called as oxygenic photosynthesis, because in plants, mainly the green plants, green is important here because let's suppose if there is a plant which does not have any chlorophyll, then it can't do the photosynthesis. So the green is very important. Or let's say if there is a plant part, for example the roots, it does not have the chlorophyll, so the root cannot show the photosynthesis. So the chlorophyll is very important for the photosynthesis to happen. And the next one, earlier the scientists used to think that the oxygen released during the photosynthesis would come from the carbon dioxide because both are gas. But no, later using the radioactive isotopes, they proved that the oxygen which is released during the photosynthesis was evolved from the splitting of water, which you will be studying in the light reaction. And let us read this. Later he envisioned a similar process of photosynthesis in plants. When glucose was observed to be a product of photosynthesis, the following equation was proposed. You have to remember this equation. 6CO2 combines with 12 molecules of water in the presence of sunlight gives rise to 1 molecule of glucose. The formula of glucose is C6H12O6 plus 6 water molecules plus 6 oxygen molecules. It is known that plants synthesize carbohydrates, the smaller and simpler ones first, and from these the more complex ones such as starch and cellulose are formed. See, the smaller and simpler ones are for example, it is the glucose. So, from the glucose, the starch and cellulose are formed. See, glucose, it is a monosaccharide. That is a simpler carbohydrate. Whereas, starch and cellulose are the complex carbohydrates. Starch, it is used for storage. So, it is called as storage carbohydrate. Whereas the cellulose, it is used in the structural components such as in the cell wall. So it is called as the storage carbohydrate. So starch, it is the storage carbohydrate. And the monosaccharide of both the starch and the cellulose, it is the glucose. We can say it this way. Let's say we have a wall. The whole wall is an example of the complex carbohydrate. Let's say starch, or it could be the cellulose. And the brick, a single brick which is in the wall, it is a monosaccharide. So the brick keeps on repeating to form a complete wall. In the same way, glucose, which is a monosaccharide, it forms the chains, or we can say it is a repeating unit which forms the starch or the cellulose. And the cellulose is the structural carbohydrate, which is found mainly in the cell walls of plant. Plants are also capable of synthesizing all other compounds such as proteins and fats, etc. First, the amino acids are formed. Later, from the amino acids, the proteins are formed. And the fats. From the lipids, the fats are formed. Animals are not capable of synthesizing carbohydrates and they have to depend on plants for the same. That is what we said. Animals show the heterotrophic nutrition, so they cannot make their own food material, which is the carbohydrates or any other proteins or fats. So they depend on the another organisms or directly or indirectly on the plants for the carbohydrates. Can we say that photosynthesis is a basic energy source for most of the living? Yes, because all the food we get is from the photosynthesis. So now let's start with the activity 1. Activity 1 is to test the presence of starch, which is a carbohydrate, which is the complex carbohydrate, and it is a polysaccharide of glucose. That means the glucose is its monosaccharide. The requirements would be, we have to take one leaf, a beaker, water, and a methylated spirit. Methylated spirit means it is the 10% methanol in ethanol. And we have to take a test tube, Bunsen burner, tripod stand. This is the tripod stand. You have to remember all these labelings. You should draw in your exam also. And asbestos gauze, iodine solution, and a petri dish. The first step includes boiling of the leaf in the water in a beaker, just as similar shown on the screen. And then we have to transfer this leaf into a test tube which has the methylated spirit. Now we have to boil this whole setup, which is a test tube containing the leaf and the methylated spirit, in a water bath. Water bath, which is can be seen on the screen, which is also mentioned in your textbook. And please remember all the labelings, and please learn to how to draw this diagram, because you may be asked to draw in your exam. And then after boiling, the leaf becomes pale, because initially the leaf was green in color. After we boil it in the methylated spirit in the water bath, all the green color goes into the methylated spirit and the leaf becomes pale. This step is necessary because we have to identify the color change during the iodine test. If it does not lose its green color, it makes us difficult to identify the color change, and the leaf loses its color because the chlorophyll dissolves in the spirit. Now transfer this leaf into a petri dish and add 2 to 3 drops of iodine or the betadine. Betadine is basically a chemical name for the compound, which is a combination of the povidone and iodine. And now if the leaf does not change its color and it is still the pale color, then it does not have the starch. And if the leaf changes its color into the bluish black color, that means the starch is present in that leaf. Now let's study the factors which are essential for the process of photosynthesis. The first factor would be the water. Von Helmont found that the water is essential for the increase of plant mass. He did not know about photosynthesis then. Later, it was found that the increase in plant body mass or material occur due to the process of photosynthesis. As we all know earlier, I have said that the food is required for the growth, repair, as well as the maintaining body temperature. So the food which is required for the growth comes from the photosynthesis. So we know the water is required for the photosynthesis. So here it says that the water is required for the increase in plant mass. Let me summarize that one. We know we require food for growth, and food comes from the photosynthesis, and we require water for photosynthesis. That means the water is the cause of increase in plant mass. Now let us study the role of air in the photosynthesis. Joseph Priestley performed a series of experiments in 1770 which revealed the essential role of air in the growth of green plants. Oxygen as a gas, that means the Joseph knew that there was a gas which was essential for the photosynthesis. He did not know that it was the oxygen, but he knew there was some gas. Later, the term oxygen was coined by the Lavoisier in the year 1775. You have to remember these years which are important. And the statement given by he performed the series of experiment. After that, the statement which was given by the Joseph Priestley is plants restore the air, what breathing animals and burning candles remove. Let me explain you. First, keep in mind that the candles require oxygen for burning. If there is no oxygen, the candle stops burning. And the second thing is the animals, if the animal has to be alive, it has to have the oxygen. If there is no oxygen, the animal stops breathing and the animal will die. And the third one is that the plants require carbon dioxide for the photosynthesis and they release oxygen during the photosynthesis. Keep in mind these 3 points, and let's see what the experiment of Priestley says. So in the first experiment, Priestley puts a single candle inside a closed bell jar. After some time, he sees that the candle goes off. It stops burning. What might be the reason? The reason might be that the oxygen in the closed bell jar is completed and there is no more oxygen present for the candle to be used during the burning. And in the second case, he also puts a mouse along with a burning candle. After some time, he sees that the mouse dies because there is no more oxygen left for breathing and the burning candle goes off. In the third case, he puts a mint plant and along with that he places a burning candle as well as a mouse. He sees that now in this third case the candle keeps on burning and the mouse is also alive. Why? Because we know that the plant releases the oxygen during photosynthesis and that oxygen is used by the mouse for breathing and used by the candle for burning. And the animal, which is a mouse, during the respiration it releases the carbon dioxide and that carbon dioxide is taken by the mint plant for its photosynthesis. So there is a continuous exchange of gases between the mint plant and between the burning candle and the animal. So this is why he says that the plants restore the air. Restore the air. Here the air is the oxygen because the plants always add the oxygen to the inside environment. And what breathing animals and burning candles remove. That is, he is referring to the oxygen here. He is saying that the oxygen is added to the environment by the plants and it is removed from the environment by the breathing animals and the burning candles. And the question that one might ask is, how does this gaseous exchange takes place? We know in animals we have the nostrils. Through that the gaseous exchange happens. The oxygen is taken in and the carbon dioxide is released out through the nostrils. Whereas in the plants we have the stomata which are present mainly in the leaves and in the other green parts. Whereas in the roots and in the stem we have the loose tissues. Through the loose tissues there is the exchange of the gases. And you can see on the screen how the stomata looks under the microscope. Let's study activity number 2, which is Moll's half-leaf experiment, and it is to identify the importance of carbon dioxide for photosynthesis. So first we have to prepare a de-starched plant. De-starched plant means the plant which does not have the starch. So for preparing the de-starched plant, we have to put the plant in a dark room for at least 3 days. Then the starch which was already present will be used up by the plant, and because there is no sunlight, there will be no further starch formed. Now after 3 days, take a wide mouth transparent bottle and put few drops of KOH, which is a potassium hydroxide solution, or few pellets of the potassium hydroxide in that bottle and insert a splitted cork in the mouth of bottle. Splitted cork which has it closes the bottle but it has a huge gap in between, so we can put some any leaf or anything if you want to, and bring this plant outside the dark room and keep it in the sunlight. Make sure now the half leaf, only the half, not the complete leaf, half leaf should be inside the bottle through the split of the cork, and the half leaf should be exposed to the air. Now keep this setup for a few hours in the sunlight. Now take that leaf, put that in a petri dish, add 2 to 3 drops of iodine or the betadine solution, and which now we have to test for the carbohydrates or the presence of starch. Now the part which was inside the bottle, on doing the iodine test, it does not show any bluish black color. That means there is no starch formed, even though the whole setup is under the sunlight, because there is no CO2, because the potassium hydroxide absorbs the carbon dioxide. Inside the bottle there was no carbon dioxide, so there is no starch formed. But where the part which was exposed to the air, on testing with the iodine, there was a bluish black color. That means that starch was formed. It has the carbon dioxide, it has the sunlight, so the starch is formed in the part which is exposed to the air. That means the carbon dioxide is necessary. If there is no sunlight, there is no photosynthesis. That means there is no starch formed. If there is no carbon dioxide, there is no photosynthesis occurring. That means there is no starch formed. Now take another leaf which was completely exposed to the air. Now check this leaf for the presence of starch by adding 2 to 3 drops of iodine. Now we will find that the whole leaf which was exposed to the air has turned into bluish black color, because this leaf was not put into the bottle and it had adequate amount of carbon dioxide present. That means the carbon dioxide was used and there were the starch formed and the leaf has turned bluish black color. So from the mole's half leaf experiment, we get to know the importance of carbon dioxide in the process of photosynthesis. Now let us study the third factor, which is light. Let me read your textbook first. In Priestley's time, scientists didn't understand about energy, but later on much was discovered about it. If energy is released when carbon dioxide and water is formed by combining oxygen with carbon and hydrogen, then what about reverse? What about forming oxygen again and putting it back in the air? Eventually, scientists learned that the energy situation would also reverse. Oxygen formation would use set energy. That means if plants form oxygen, they have to get energy to make it possible. Where did the energy come from? So the first, he is talking about the respiration. In respiration, be it carbohydrates or proteins or fats, anything which has carbon and hydrogen, it combines with the oxygen to give rise to the carbon dioxide, water and the energy. So during the respiration, energy is released. If we just reverse the scenario, that means if from the carbon dioxide and water and the energy is used here, it gives rise to the carbohydrates mainly and other during different process. This would be called as the photosynthesis. So just if we reverse just the process of respiration, if it is reversed, then it will cause the photosynthesis. Now they are asking, where do we get this energy from? Then this energy which is used during the photosynthesis comes from the natural sunlight. In 1779, a Dutch scientist who was a Jan Ingenhousz, he did an experiment to find out the importance of sunlight in photosynthesis. So he did an experiment in which the bubbles were formed and those bubbles were of the oxygen, and he found that this oxygen was released only in the presence of sunlight. If there was no sunlight, there was no oxygen bubbles formed. So let us study about his experiment. So for the hydrilla experiment, we required 2 beakers, 2 funnels and 2 test tubes and 2 plants, hydrilla plants. So we have to take a test tube, insert a funnel in that and put the hydrilla plant just as shown on the screen. Now you have to reverse this whole setup and put that inside a beaker which was filled with the water. Now we have to prepare 2 similar setups. One setup has to be put under the sunlight and other one inside a dark room. So in the apparatus which was placed in the sunlight, here there were the oxygen bubbles formed and on the top there was the oxygen getting accumulated. So when we put a glowing matchstick or the incense stick near the oxygen, the matchstick or the incense stick will burst into the flames in the presence of oxygen. So when they already glowing matchstick or the incense stick was born to the gas present on the top, it was bursting into the flames. That means the scientist has known that it was the oxygen gas accumulated. This happened only in the presence of sunlight, because in the presence of sunlight the photosynthesis has occurred and the oxygen was released. But whereas in the dark there was no sunlight, so there was no photosynthesis happening. That means there was no oxygen released. Here all there was the water. So this was the experiment of the engine house. From the engine house experiment, it has been concluded that the sunlight is necessary for the photosynthesis. If there is no sunlight, there will be no photosynthesis occurring and there will be no oxygen released. So coming to the third activity, here also we are doing this experiment to know the importance of sunlight in the photosynthesis, but here we are checking for the presence of starch. But in the hydrilla experiment or the engine house experiment, we did it by testing for the oxygen release. So coming to the experiment, we need a black paper and a plant. So take a black paper and make it any design on it and cut through the design, be it S or anything, just cut through it. Let's say this is a black paper and you are doing some one design, and this all should be the black paper. You have to make a cut in it, and when you do that and place it on a leaf, so when you place it on the leaf, here, here and here, the sunlight will be falling on that leaf. But where there is the black paper covered, on this part there will be no sunlight falling on it. Now we have to leave this apparatus in the sunlight for some time, and after that we have to take that leaf, remove the black paper, and then you do the iodine test. That put that leaf in a petri dish, add 2 to 3 drops of iodine and check. So when we do the iodine test, the part which was directly exposed to the sunlight, that means the S over here and these 2 parts, here there will be the color change. These 3 parts will change into the bluish black color, showing the presence of starch, because these 3 parts were exposed to the sunlight. But here where the black paper was covered, there is no color change. It is only the green in color. That means here there was no starch formed, because this part where the black paper was present, there was no sunlight falling. So from this experiment we know that the sunlight is necessary for formation of starch also. So let's talk about the fourth factor, which is the chlorophyll. Engine house wanted to find out more about photosynthesis and carried out several other experiments. He proposed that only the green plant parts could carry out the process of photosynthesis. What about plants having the colored leaves? See, if a plant has the red or yellow leaves, if the leaves are not green in color, so in that case also the leaves can show the photosynthesis, because they still have the chlorophyll which is hidden under the other pigments. They can still do the photosynthesis even though if the green color is not clearly visible. So how is that new leaves which look dark red in color in several plants turn green? See, initially the new leaves are usually red or other color. In that leaves, the anthocyanin pigment, which is the accessory pigment, is more. And when the leaf matures, those leaves turn into green because the chlorophyll production increases. So we can write here due to the increase in chlorophyll production. Whereas do plants having reddish or yellowish leaves also carry out the photosynthesis? Yes, because they have the anthocyanin pigment. Let me write over here. So the leaves which are yellow or reddish also show the photosynthesis because they have the anthocyanin pigment. What made plants carry out photosynthesis while green colored animals like birds could not? See, the plants have the chloroplast, or which has in turn the chlorophyll. So they do the photosynthesis, whereas animals, even though if they are green in color, they do not have the chlorophyll or they do not have the chloroplast. So in any case, the animals cannot do the photosynthesis. Establishment of engine house proposition became possible in the year 1817 due to the work of two scientists, Pelletier and Caventou. They obtained an extract of the green colored substance and named it the chlorophyll. It was found that the pigments, in addition to the chlorophyll, carotenoids and the phycobilins, could also aid in the process of photosynthesis by passing on the energy of sunlight trapped by them to the chlorophyll. So these pigments which trap the energy of the sunlight and pass it to the chlorophyll, then these are called as the accessory pigments. So yeah, that's it for today. The remaining chapter will be covered in the part 3. Subscribe for more.